Good afternoon, everyone. This is Anthony Alaka with Torrey Hills Capital. I want to welcome you to the RxMP Therapeutics webcast for November 3rd, 2021. I hope you're all doing well, and I want to thank you for attending today's virtual presentation. We're very excited to bring you RxMP Therapeutics. Excessive bleeding associated with trauma, surgery, or bleeding disorders is one of the most common preventable causes of death. RxMP Therapeutics is developing a first-in-class systemic hemostatic agent for the treatment or prevention of hemorrhage and excessive bleeding. The company's lead product, RMP-402, has shown solid preclinical data in controlling excessive bleeding regardless of cause by using naturally occurring red cell-derived membrane particles, or RMPs. The company is addressing a market that's roughly $5 billion. RxMP is in the process of a crossover round of financing, the purpose of which is to capitalize the company through preclinical development and the filing of an investigational new drug exemption, or an IND, while preparing to conduct the first in-human safety trial for RMP-402. With us today to discuss RxMP Therapeutics and the company's strategy going forward is CEO Rif Pamukcu, and we also have Dr. Mark Swaim, editor of BioPub and our resident expert on all things biotech and pharma. It's great to have you both on today. I just want to, before we get started, say that if you have a question during the presentation, obviously the easiest way is to simply type it into the chat box or the Q&A box, and we'll make sure it gets answered. I'm going to turn it over to Dr. Swaim for some opening comments. Then Rif Pamukcu will take us through the investor presentation. Then we'll open it up for Q&A. So with that said, Mark, the floor is yours.
Thanks, Anthony, and I appreciate the opportunity to be here with Dr. Pamukcu and you and talk about this remarkable pre-market investment. As you know, this early stage investing can be potentially very lucrative. I don't need to explain that to this audience. And as a career-long bench and clinical coagulation person, I love this company because I've never seen anything like the agent that they're promulgating, an IV first-in-class, universal, agnostic procoagulant drug with no apparent risk of provoking off-target clotting or undesired clotting. Never been anything like it. Now, mind you, if you have a hole in your femoral artery aorta, probably not going to control that. That's a surgical problem. But for non-surgical bleeding, which can occur for a panoply of reasons, this is a very useful agent, appears to be. It's extremely promising, and I'm very happy to be involved in trying to get it through the development stages and to market. And I want to limit what I say today to allow our main guest, Dr. Pamukcu, plenty of berth to present all the slides and really discuss it at length. I think it's a wonderful opportunity. For sake of full disclosure, I'm invested in it as well. Rif, good to see you, and thanks for joining us.
Thanks. So let me share my screen here, which, given that we do this 10 times a day, you'd think that it would be easier.
Full screen, Rif.
You got a full screen now?
Yes.
Now we do. Good. Looks
Got
good.
it. Okay. Yeah. All right. Thanks, Mark. Thanks, Anthony. I'm glad to see all of you, though I can't see you attending here. But as Mark said, RxMP is developing a first-in-class therapeutic for excessive bleeding. And let me just get through the forward-looking statement slide, which is, I consider a Where's Waldo kind of slide. We change it from periodically and see if anybody notices the difference. But anyway, I think this is a sort of a unique investment opportunity. Obviously, I wouldn't be spending my time and energy on it if I didn't believe so. But I think it's an attractive risk-reward profile in the field of hemorrhage, which, in the investment communities, gene therapy is sexy, immuno-oncology is sexy. Bleeding is not sexy, but bleeding is a major still a major cause of death in this country, Europe, Japan, rest of the world, et cetera. But we see a multi-billion-dollar market expansion opportunity that starts with a small investment to very solid clinical milestones that also includes an orphan indication that can get us to the market quickly, yet then be able to show the data to hit larger expansion markets. We've got experience in management and advisors. I'll cover some of that at the end. But myself, I'm a gastroenterologist by training. Founded my first biotech company when I was a GI fellow, Cell Pathways, took it public, got it acquired in a bidding war between a couple of pharmaceutical companies. And I'm a glutton for punishment. I do this sort of thing over and over again now. So when this platform technology was introduced to me by the Wallace H. Coulter Foundation at the University of Miami, I was intrigued. It was headed towards developing a product for hemophilia, but it really didn't feel like that was the appropriate indication for it initially. It will help bleeding hemophiliacs, but it will also help a much broader market of bleeding patients. It's a platform technology in that these are red cell derived membrane particles. And I'll describe them a little bit more, but you see them in the vial with the microparticles here on the right side of the screen. These membrane microparticles are naturally occurring in the bloodstream and assist us in the process of coagulation. But that's been a more recent understanding. And we're capitalizing on this. These particles here are not extracted from humans, but made from blood that's approved for human use. But we manufacture it outside the body, so we can manufacture quite a large number of these at any given time. So this is a fully scalable process. We've got worldwide patents issued, and we've just got a new composition of matter patent that was recently allowed in the United States and is still going through prosecution elsewhere. But this is a good harbinger for this new set of patents, and this new set of patents will expire in 2037. So we've got long running room here to the market. Our RMP 402, which is what you're seeing here, is the lead product. I'll show you a little bit of the solid preclinical data we have and show you how we have a sort of a clear initial clinical path to results, proof of concept, value inflection points, et cetera. We're very capital efficient, semi-virtual, value inflection points, I believe, in 2022, 23, 24, with a potential NDA filing in the orphan indication in 2024, coming to market potentially in 25. So a fairly rapid program. So clinical bleeding, we've got 3.5 million people that are being treated annually for bleeding, not anemia, but active bleeding in the US and Europe and Japan. 30 to 40% of deaths due to trauma and crush injury are actually still from hemorrhage. And that's accounted for about 150,000 annually. Hemorrhagic stroke, our orphan indication, along with the parallel clinical studies in the larger one, 40% of those individuals hit the emergency room bleeding and are in a good position to potentially respond to our agent. Often we see excessive bleeding in the surgical suite, obstetrical hemorrhage, major GI bleeding. These are all major markets for us. The market potential is 5.7 billion and going north. And I'll explain how we might go north a bit later. But even our entry markets between the treatment of acute brain hemorrhage to prevent the expansion of the bleed, an orphan indication is got a $600 million market, and spinal fusion, our initial market for demonstrating the effect in excessive bleeding in surgery, by itself is a $900 million market. These patients, when you fuse 3 or more levels of the spine, lead to greater than 20% blood loss from a large number of patients on the table, to the point where they try to recapture their cells and with cell capturing devices and try to give them some of it back after the OR, which has incidence of infection and other morbidities associated. but also leads to increased morbidity and cost. There have been medical economic studies in spinal fusion showing if you lose more than 2,000 cc's or 2 liters of blood on the table, you have a much higher cost in terms of ICU stage, much higher morbidity. So unlike a lot of other products, the medical economics analysis has already been done here if we can reduce the amount of blood loss in surgery. The expansion markets are another 4.2 billion and up, and I'll show you some numbers on that in a bit. And the current therapies are truly inadequate. Competition is limited, but I think recombinant factor 7A, which is the 800-pound gorilla amongst those other therapies, gives you a sense of the unmet need. This agent was approved for a rare form of hemophilia. It's anywhere between 70 to 95% of their sales are off-label, even though they have severe adverse events with a $1.5 billion market in the excessive bleeding market. So I think it gives you a sense of the real world market as well as how much unmet need there is perceived out there. So, homeostasis in us, hemostasis in us is a very, very finely tuned process between functions that we have that promote normal clotting, adequate, procoagulant factors, adequate platelets and fibrinogen, balanced against things that put brakes on clotting or break down the clots too quickly to lead to excessive bleeding. What's been underappreciated is that the role of phospholipid membranes in the clotting process. And RxMP is capitalizing on this aspect of it. All of the other pieces in this equation have been addressed through various products. It's the phospholipid membrane piece that, like I said, is underappreciated by the market and gives us a market opportunity. So clotting tests, whether you're in the heat of battle with an acute bleeding episode in the emergency room or you are prepping to go into the OR, they give actually quite limited guidance. Coagulation tests like the thrombin time, PTT, et cetera, they're only really abnormal when the factor levels reach 15% of normal. So things that kind of in between don't tell you much and don't give you a lot of guidance. Sophisticated tests looking at how quickly a clot breaks down take time, usually are not helpful in the acute setting and aren't available in the vast majority of hospitals. Pre-op bleeding tests such as the bleeding time are reproducible, inaccurate, often are done, but don't really predict who's going to excessively bleed in the OR. So when a patient comes in bleeding or is bleeding in the OR, the clinician's options are quite limited at this point. We've got various forms of fractionated blood products. We've got procoagulant agents. We've got antifibrinolytic agents or agents that try to prevent the clot breakdown. Either of these things are limited in use because of restrictions put on the physician by the hospital systems, or they've got adverse events, prohibitive costs, et cetera, complications like off-target clotting. So in some cases, even allergic reactions to the product. So the armamentarium, while it's there, is not particularly good to address the fundamental problem. We tend to stem the tide by giving more blood. We don't have a lot of things that are really good for stopping the bleeding itself without limitations and adverse events. So, where does that leave us? Where does that leave most doctors? Most doctors are pretty uncertain how to proceed beyond, what they know about trying to stem the cause of the bleeding, find the site, coagulate it, sew it up, et cetera. The diagnostic workups usually on the fly are non-diagnostic and don't lead you in a particular direction. And most hospitals don't have a coagulation medicine consultative team to help out. So we saw an opportunity how to promote good hemostatic activity safely. And so a new concept started to emerge about 15 years ago, where the founding members, Dr. Zian An, Wen Shi, started working in the area of microparticles circulating in circulation. And what they found is that compared to platelet microparticles, white cell microparticles, endothelial cell, which is the endothelial cells, the lining cells of the blood vessels, microparticles from those tissues. Those microparticles have a fundamental problem. They cause rampant off-target clotting. So while you may try to give them IV, you're going to end up with strokes, you're going to end up with clotting in coronary arteries, you're going to end up with blood clots in the legs that will embolize. Right. So those have not been a good approach in the particle space. What they did discover is that the RMPs, the red cell membrane microparticles, are actually a naturally occurring solution. They provide phospholipid membrane as a therapeutic agent and have been demonstrated in clinical studies that the higher level of RMPs floating around in your bloodstream reduces your likelihood of bleeding, particularly in cases where you have platelet dysfunctions and the like. And if you do start bleeding, the higher levels circulating in the bloodstream demonstrate that they have a reduced number of units needed for transfusion. So how do we go about providing these particles? Well, this is where RMP 402 comes into play. These are manufactured under high pressure and mechanically from packed red cells, again, approved for use in humans to begin with, lyophilized down or freeze-dried down. And you see the freeze-dried product there. This can be reconstituted with sterile saline, sterile water injected, in our case, in the initial studies in animals, and hopefully eventually in humans, and will dramatically reduce the bleeding and stop it within a matter of minutes. So what I'm showing you here is the clotting cascade that involve the various clotting factors that lead to clot formation along with platelets. What you will notice with the green ovals, that phospholipid membrane is integral to all of these steps. And that is the piece that hadn't been really appreciated until recent history. And we are now providing that phospholipid to this whole cascade. The red cell membrane is asymmetrical. The outside of the membrane is composed of neutral phospholipids and don't promote any kind of bleeding, clotting. The inside of the membrane has phosphatidylserine. And phosphatidylserine turns out to be an important anchoring point for many of the factors involved in blood clotting. So when these phosphatidylserines are out there, they provide scaffolding, if you will, for the clotting factors to come together, for the platelets to come together, and it synergizes the overall activity. So if you inject these red cell membrane microparticles into an animal or a human, they will circulate throughout the system. They don't have something attached to them called tissue factor, which is what causes the off-target clotting seen with other microparticles. They will attach themselves to the bleeding site where the blood vessel is broken and the collagen is exposed. They'll attach themselves like Velcro there, and then they will actively synergize the clotting factors and platelets locally in this scaffolding network to rapidly induce the clot formation. So where is RMP 402? Well, we manufacturing it using a high pressure extrusion method, which involves, as I often describe it, as putting them under extremely high pressure, blowing it through the red cells through a pinhole, slamming against the metal plate, and breaking them down into very uniform 1 micron microparticles that are partly right side out, partly inside out. And what we discovered, and was the subject of our newest patent in the composition of matter patent, was that there's a sweet spot in inside outness versus right side outness. If you go to either extreme, you go to fully right side out or fully inside out, you dramatically lose the activity of these membrane particles. So we've captured the way to manufacture these and manufacture these in a very consistent batch to batch approach. You see a small tabletop version of one of the devices, a cell disruptor that we use. This is fully scalable to industrial levels. Actually, these cell disruptors are used predominantly for harvesting proteins and the like from bacteria and yeast. We've managed to seal these up, make them sterile, et cetera, for this purpose. We are GMP ready. We are scalable. We can make it consistently. We have a lyophilized sterile product at the end. And we've seen in preliminary stability studies of a year to 18 months, beyond, we can keep these products stable for 18 months, refrigerated or frozen. Packed red cells, as a reference, are only good for 42 days. So the idea is, if you administer one of these units to an individual, you could dramatically reduce the number of units of blood products that are used in patients. In sometimes in surgery, 10 units plus could be prevented by a single dose of this. In trauma patient, we're talking about hundreds of units that they might receive if we can get them stabilized and clotted before they have to use a lot of product. We're saving a lot of blood, platelets, and the like out there. Our plans were vetted with the FDA in a Type C meeting. We've got a good roadmap for the non-clinical manufacturing and clinical portions of this. We're planning on having a pre-IND meeting in the upcoming months to finalize a few aspects of this. But we've got like I said, we've got an excellent roadmap from the FDA in our discussions with them. So this product itself, and you can kind of see it here, as I mentioned, kind of going to the site of bleeding and act as the scaffolding there, is administered IV. It targets only the site of bleeding. There's no off-target activity. We've done initial animal tox studies, GLP level tox studies in rodents at this point. Our initial IND enabling tox studies. We've seen no off-target clotting, brain, heart, lung, other tissues. And we've seen that in something called a thromboelastogram, where we can put in human samples that are treated with antiplatelet drugs or have genetic diseases such as hemophilia, or have other platelet disorders. Under all circumstances, when you put in the RMPs, you normalize the coagulation and platelet functions back to normal. Right? So this is, in a sense, a universal procoagulant. So we've got a number of different animal models across species that have shown the same result. I'm showing you one of the most rigorous of these is a rabbit liver trauma model. You lacerate the liver of rabbits in a very prescribed way. As you can see with the black line, they try to exsanguinate and die within a matter of 2 hours. If you administer the RMPs to the rabbit IV after the bleeding has started, you'll see about a 30 to 40% reduction in blood loss and a higher survival rate. All right. We've seen this in chemotherapy-induced low platelet count models in rabbits, tail vein laceration models in rats, rats treated with antiplatelet drugs, and more recently, the neurology division of the University of Miami got a $1.5 million grant from the NIH to study our project in hemorrhagic stroke with very similar results. And if you look at this set of panels, these animals had a agent called collagenase injected up their carotid artery, and it breaks down the blood vessels, and they start to bleed into the brain. And as you can see, whether from the control to a third of the dose to a full anticipated human dose, the magnitude, the size of the clot, of the blood clot is much lower. The green bars in the middle actually show you the numeric values across the whole brain with a statistically significant reduction. And the far right shows you the recovery time. And it's well known in humans that morbidity and mortality is associated with the size of the hemorrhage. So you can see these animals recovered a bit more quickly. And, I attribute part of that to the fact that they started with a smaller clot size overall, but they recovered at a reasonably brisk rate. So this has led us to adding hemorrhagic stroke, which is an orphan indication, as one of the clinical indications that we'll have in the plan going forward. So, and by the way, that was given an hour after the animals started bleeding, which is about the same time that a patient would roll into the emergency room. So what are the distinctive properties of the RMP? So we know that it amplifies, sort of a tissue factor independent blood coagulation. We noted that it enhances platelet aggregation in and around those coagulation factors. But more recently, we've also seen that it has antifibrinolytic activity. In other words, it helps stabilize the clot from breakdown. We've also seen some anti-inflammatory activity in the hemorrhagic stroke, and inflammation in stroke also increases morbidity. So that's actually good news as well. So this leads to a very, very broad spectrum of potential applications. So look at markets for a moment. In the initial orphan indication, about 40% of these individuals hit the emergency room bleeding. They would be the ones that would want to treat, and our clinical trials are already being designed around that population. Not treating all comers coming in, but the ones that are showing on CT scan in the ER that they are still actively bleeding. Because obviously, if they've stopped, we're not going to be able to demonstrate a clinical effect. So we can enrich our clinical trial protocols with the active bleeders. Orphan drug pricing is available here. And as many of you might know, orphan drug pricing can lead to, hundreds of thousands of dollars. But we've taken a much more conservative stance on calculating this market potential. TPA and similar products are used for thrombotic stroke at about $8,000 per dose. This is a disease, brain hemorrhage, that's going to have some significant morbidity no matter what you do. So we might not be able to go to those lofty price ranges. So we use the TPA pricing as just as a initial conservative indication of market size, leading to about a market potential of about 600 million US, Europe, and Japan at this point. The initial market expansion into the patients that have lumbar surgery of multiple levels, again, about 40% of these patients require a significant amount of blood products, in many cases 10 or more units. But you don't know who this is going to be coming into the OR. So each of these patients would be treated prophylactically by the anesthesiologist. As the surgery begins, they would be given this IV, a third of the dose initially, and then the rest of the dose run over the course of the first hour, with an average cost at about, giving blood products is an average cost of about $1,700 to $1,800 for these patients, not including the cost of adverse events, et cetera. And we're using that as our benchmark for this population for predicting a market potential of 900 million. If we see, really excellent efficacy with this, pricing may actually go higher. Just for your point of reference, a single unit of blood administered fully loaded is somewhere between $800 to $900 a unit cost to the patient. So the expansion opportunities are here: trauma, GI bleeding, orthopedic surgery, obstetrical bleeding, cardiovascular surgery, other surgical medical complications, surgical oncology. Just these markets where we believe we can make inroads after the excessive bleeding indication. And we're not going to be going to the FDA asking for excessive bleeding in spinal surgery. We're using spinal surgery as the model for excessive bleeding. So we're hopeful for a broader package label, and we can expand fairly rapidly into the use into these much larger markets. Now, our phase one, normally a phase one result is kind of baked in to most considerations, but our phase one actually has the opportunity to lead to even further market expansion. Right now, we're talking about those individuals at high risk for excessive bleeding, brain hemorrhage, in the expansion markets. And if we, in our target product profile there is that we're going to demonstrate that we reduce brain hemorrhage and surgical blood loss, that there will be a reduction of need for blood products in the surgical suite, that we're non-inferior to packed red cells, but will actually be safer because we will need to do viral clearance on our product, because our product is allogeneic. It's not manufactured from a single unit. It's manufactured from hundreds of units of packed cells. So once you get above 10 units, we need to do a viral clearance steps in the manufacturing process. So this actually makes it safer than receiving the multiple units of packed red cells. And the convenience would be that this would be available in the ER, in the OR, and actually potentially eventually move out into the rescue squads, helicopters, et cetera. Now, the one thing that you do by introducing all of this small amount of membrane is there are lots of minor antigens there. This will be, type specific, RH specific, but there are a lot of minor antigens there. And if you develop what's called alloantibodies to those minor antigens, it'll make it a little bit more difficult to type and cross, but it doesn't change the safety of receiving blood products in the future after you receive this, because you won't develop any significant hemolytic reactions there. But this product is only in circulation for 15 minutes. This product loses its activity after 3 hours. We know, particularly in this COVID era, that it takes days to weeks before you develop an antibody reaction. We don't believe, and we don't believe the FDA believes, that we will develop antibodies to, or patients will develop antibodies to these minor antigens because of the extremely short exposure. If that's the case, then this actually becomes safer than giving multiple packed red cells in surgery, and we could see an expansion of the market beyond the 5.7 billion. We can measure the amount of circulating red cell membrane microparticles. We're not a diagnostics company, but a Quest and a LabCorp, et cetera, could easily set this up it's a flow cytometric assay, and it could become a standard pre-op test to determine whether average individuals, are at excessive risk for bleeding in the OR. So we could really see a much larger expansion at the end of phase 1, depending on the phase 1 results. And the phase 1 results we'll be looking for is alloantibody production. So this is the clinical design at this point. We're raising this money to pay for the purple, get us to the IND, do GMP manufacturing on the material, and finish out our IND enabling safety and farm tox. File an IND the latter half of 22, go into phase 1. And our discussions with the FDA was to go into a minimally invasive group of these spinal fusion patients. So these patients would be getting 1 or 2 levels done rather than 3 or more, which is the larger group that would be used in the phase 2A. We would look for alloantibodies in the beginning. We would administer a dose to them in an ascending dose study. We would, after about 4 weeks, and this is an elective surgery, after about 4 weeks, we would look for alloantibody production. They would go into surgery, they would receive another dose, and after another 4 to 6 weeks, we would look for alloantibody production. The FDA actually, during the meeting, said this is one of the most innovative phase 1 designs they've seen in a long time, and that in one single study, we satisfied their requirements for single and repeat exposure in patients, and we could then, if demonstrated to be safe, could then proceed into our phase 2 program. And our phase 2 program would be parallel studies, one in the bone, in the brain hemorrhage individuals coming into the OR, and this could be an 80 to 100 patient registration trial. Could get us to an NDA under the Orphan Drug Act, potentially on just one single trial. And in parallel, do a spinal fusion study in approximately 100 to 120 patients, in patients that are undergoing 3 or more levels where they're likely to lose 2 liters or more of blood, and use that as a phase 2A result to demonstrate the proof of concept in the larger indication. Success in either one of these trials would likely mean that probably acquisition by one of the very much larger players. Though, as a small company, we in fact could take this to the market for the orphan indication. All of this being done on approximately $12 million total, including the first $3 million that we're raising now. So of the first $3 million, this gives you a sense of the use of funds, mostly manufacturing, animal tox, and prep for the phase 1 study. And we would be looking at raising additional monies as we become a clinical stage company with a bump up in the valuation to support the clinical programs. In terms of experience, I, of course, am the CEO. I took Cell Pathways public, as I mentioned, got acquired in a bidding war between OSI Pharmaceuticals and Lilly. OSI actually won that bidding war. Wenxi Ji is one of the inventors and is the chief scientific officer of the company. Sue Van is also a director, and she is the chief executive officer of the Wallace H. Coulter Fund, which is one of our leading investors that initially funded this. In terms of advisors, we've got industry level advisors in Jim Nolan coming out of Wyeth Nurse, and Mike Fair also out of Wyeth. Toby Silverman, Ashok Mukherjee are advisors, and I took them to the FDA with me. They are both well known to this particular division of CBER, the blood products division of CBER, and which served us well. In terms of scientific advisors, we have Yian An, who is also one of the co-founders and inventors of the technology. Gerald Levy, who's in surgical intensive care. He is an anesthesiologist and a hematologist at Duke. Sam Schulman, who's recognized world-renowned in thrombosis out of McMaster in Canada. Barth Green, also recognized worldwide in neurosurgery at the University of Miami. Kunjan Dev has been the professor doing the work on the brain hemorrhage at the University of Miami. And Dan Holler is a recent addition to our scientific advisor board. He's a trauma and critical care surgeon affiliated with Mount Sinai and Mercy Hospitals in New York to give us the trauma surgeon perspective. Even though we won't be going into trauma first, because those are very, very difficult clinical studies to do, we want to ensure that we've got all of our ducks in a row so that this can expand into the trauma setting after it reaches the market. So we've had conversations already with some of the biggest players, particularly the ones in the left column. Bayer expressed interest, as did several of these others, at seeing the phase 1 data. Bayer likes to be on both sides of the coagulation platform in terms of drugs. They actually owned aprotenin that they eventually sold off to a smaller Swedish company when it got black boxed and in some cases removed from the market because of allergic reactions. It was one of the few procoagulants out there. CSL and Behring and Baxter and Grifols are all in the blood space. Baxter actually was an interesting story. When I met with their team, their first question was, well, how do you extract this from humans? And when we told them the story that we don't extract it, we don't purify it, we manufacture it, was on one of those hand slap to the forehead statements of why didn't we think of that? Grifols and CSL are not common names in terms of the drug industry, but they happen to be the world's largest suppliers of fractionated blood products. They've got the supply chain, since we make this from pack cells. They understand the distribution channels. They understand mechanical manufactured products in the blood space. Grifols alone does $6 billion worldwide in fractionated blood products, and they got this very, very quickly and are very interested in seeing the phase 1 data. Ashika Kasai has also expressed interest out of Japan. Novo Nordisk owns recombinant factor 7A that recently came off patent. Octopharma is alike a Grifols and a CSL, as is LFB at a smaller stage. The rest of them all have hematology-based programs. So I think we have a number of interested parties that are keeping an eye on the company as we move forward in our development program. So why invest in RxMP now? I think we've got potential exits coming in terms of acquisition or IPO as we enter the clinical arena. The end of phase 1 will lead to a value inflection point, proof of concept in clinical data, yet another major value inflection point. We've got large markets, an orphan indication to get us into the markets a bit more quickly, clear and measurable clinical endpoints in reduction in blood loss, capital efficient in terms of being able to do this on actually very little money as compared to others, a platform technology that we haven't even capitalized on yet because we've been resource limited, which includes further modifications of these RMPs to target specific high-end needs. That is a distinct possibility. We've got worldwide patents issued in major markets, and we've got a team that's been there and done that in myself and the advisors that can get this thing to these endpoints. And so with that, I can feel free to go to questions.
Yeah, thank you, Rif. Appreciate it.
Fantastic.
Obviously, a lot of great things happening with the company, and I think this is setting up to be a pretty interesting situation here with everything you have going on. We already have some questions in queue, but I just want to remind our attendees that if you have additional questions, just type them into the Q&A box, and we'll make sure they get read or answered. So, Rif, first question. So the slide that talked about the potential market size, on the bottom left, the date you cited is, it says 2004. Can you give more detail on the sources and basis of, your market need estimate? What about existing treatments? What market share percentage are you including in your estimates?
Let me pull that one back up. We're talking about this one.
He's talking about, let's see here, with the potential market size. I think it was like this.
1970.
A little bit. Yeah.
I think it was at the beginning of the actual presentation.
Oh, this one here. Oh, so that 2004 study was the medical economics study in the spinal fusion patients, right? So it's sort of unusual to see medical economics studies done, but this one, like I said, this one was sort of unusual in that they looked at spinal fusion and tried to understand what the cutoff was so that they could try to direct intervention in it. And this is where they use the cutoff of, or analyze it to a cutoff of, above or below 2 liter blood loss in the surgical suite. These numbers, because of the expense, these numbers have probably only gotten worse rather than better in terms of morbidity and cost, and we might even see advantages at even a lower loss today than back in 2004. But that's the only available data on this piece in terms of that. And one of the thing about it is I'm fully cognizant that when you're doing those sorts of clinical studies, if you feel like you need to make the case to get insurance, reimbursement, and for drug therapies, medical devices often, you have to jump through a lot of hoops in terms of the reimbursement. The real issue becomes with drugs is, how much is the copay? The drugs, and this would be considered a drug, therapeutic, gets paid for. The question is at what level? In this case, to the hospitals, because the hospital would be the one buying it, or the rescue squad company would be the ones buying the product. But I'm fully cognizant of often the need for those medical economic analyses. And when you're doing your clinical studies, it's not uncommon to spend a few $100,000 extra during a clinical trial to try to capture that medical economic data to kind of refresh the information that's available, or provide new information to support your reimbursement aspects of it. So that's where that 2004 citation comes into play. The rest of it is, when we get into these sorts of numbers, in terms of lumbar surgery, the numbers in terms of surgical numbers, et cetera, span from 2007 to 2017. The average cost per patient in terms of blood consumption is based on numbers out of the Red Cross and similar entities that provided numbers, for the use of blood products, the average number of units, et cetera, in various indications, and that's how we got to these numbers as well. There have been, in this case, this was the American Blood Centers did an analysis of how much blood was being actively used in all of these various indications. So that's how we came to our numbers. We're talking about for everything other than the brain hemorrhage, we're talking about a $1,700–1,800 cost per unit, price per unit. Our costs are obviously substantially lower. And we know that as we go to scale, that we buy pack cells from the various groups, the Red Cross, other distributors for manufacturing. But as we go to larger scale on this, our cost for those pack cells is going to end up being, somewhere between a half to a third of what we currently pay at research scale for the blood. So there would be a very wide margin on this product, even at $1,800 a unit, and that would come into sort of parity with giving 3 units of blood, 2 to 3 units of blood in the OR.
Gotcha. Okay. So to piggyback that question, what about existing treatments? And if so, how much market share are you looking to grab from any current treatments? I know that was on one of the slides in terms of the other treatments that are available, but can you describe, what the plan is?
Well, I don't think most of these things are, fresh frozen. It's harder to quantify these. They're really kind of used in specific settings. I mean, the blood products like pack cells, et cetera, you're going to be pouring, theoretically, if say a trauma patient comes in, you're going to be pouring pack cells into them and potentially other factors to try to normalize them in the process while you're using our product to try to stem that tide. So those aren't, those are kind of synergistic here. It's not really a competitive agent. Vitamin K, you only use those in vitamin K deficient patients or patients on anticoagulants, like Coumadin. Though, it's interesting that in a setting like that, if a patient rolls in, needs emergency surgery, is on an anticoagulant, while they might try to reverse them with vitamin K, and then have this problem where they're going to have to be sitting in the hospital for days to get them back onto the anticoagulant. This product looks like it can be used to transiently give them the ability to coagulate during surgery, and the moment they're out of surgery and our product's off, they go back to their previous state of coagulation. So that would dramatically reduce the use of vitamin A as a competitor there. The other ones like desmopressin, they're very limited uses because of restrictions, because of adverse events. Many of the antifibrinolytics, same sort of story. They've got off-target activity. The physician's hands are often tied in terms of when and where they can use them. Recombinant factor 7A is prohibitively expensive. It's like $10,000 a milligram. So they're using it, but they're using it in essentially very, very extreme cases of massive bleeding in the trauma setting. I think that we can eat into that significantly, I believe, and in terms of the $1.5 billion market that they have there, because I think our product will be significantly safer because we don't have that, what, I've used Mark's word here, the coagulation overdrive that occurs, and the substantial risk of off-target clotting brain, as well as particularly clots in the lungs, in the legs that will embolize. So I think we can make significant inroads there. The rest of them are kind of smaller markets. We're creating a fairly large market unto ourselves at this point.
Yeah.
I think in to some extent, what we're probably going to eat into as well is there's going to be a reduction in units of blood used. All right. And in all honesty, that isn't the big driver for the blood banks. They don't make a whole lot of money on the pack cells. When they get blood donations, where their focus is financially is primarily the platelets, which are a high margin product for the Red Cross and others. We will probably add to the availability of blood by reducing the number of units used, particularly in settings where there's sudden high usage, catastrophes, and the like.
Okay, great, thank you. I want to try to get to all these questions.
Sure.
Is RMP 402 product different from that approved product used for hemophiliac bleeding? If so, how?
Well, there are no RMPs approved for hemophilia. The agent, other than some factors that are being used for hemophilia, where the various hemophilias where you're replacing the factor itself, recombinant factor 7A is a clotting factor, right? So it's a protein product and a recombinant clotting factor. So the red cell membrane particles are completely different technology. In theory, they could be used together, but if we're successful, they're really not going to need to use the recombinant factor 7A in the off-label indications. That would be relegated then to just the hemophilia indications that they're approved for.
Another good question. Does Coulter have a royalty on RMP 402? What is the extent of their involvement when it comes to that?
Coulter is a philanthropic organization primarily. This was made as an investment from sort of their philanthropic side, is a sort of a mission related investment to them. They do not have a royalty. They have an equity position in the company like any other common stock shareholders. All stock at this point is common. There is a very, very small royalty back to the University of Miami for this, low single digits at this point, which is standard when you're taking a technology out of a university.
Gotcha. Are these RMPs used just broken up human RBC membranes? Do you have to match it with patient blood type to avoid an adverse reaction, or are RMPs made from RBCs from universal, quote unquote, universal donors?
Currently we make them from O positive. So that you'd be able to actually use it in clinical trials, et cetera, for anybody who is Rh positive. When this finally comes to market, we believe that we will have multiple SKUs. They will be type specific in terms of, A, B, and O. They will be Rh specific, though you will never have O negative because that's, you'll never get access to O negative blood for manufacturing because it's used in trauma and other circumstances. It will be Kel negative because Cal is one of the few things that we screen for, because Cal, if you develop or have Cal antibodies, could lead to a hemolytic reaction. So we don't want to induce Cal antibodies in people that are Cal negative, and that's about 85% of the population. Regarding the other minor antigens, it will not be typed for that. So when this will be used, obviously in sort of unknown situations, it would be O positive. In other known situations, in the surgical suite where you actually know the patient's blood type, you can go to the specific type. Now, what happens if you incorrectly gave it, right? So what would happen is that if you, for example, gave a B to an A individual, you would just simply rapidly clear this. These particles do not contain any hemoglobin. There's a little pink tinge to it, but that's very, very trace amounts of hemoglobin. But unlike PAC cells, this doesn't have any hemoglobin to speak of in it. So even if you ruptured them, you would not be dumping a lot of hemoglobin out, which in a transfusion reaction would lead to injury to the kidneys, for example. Right. The most that you would see here is a loss of efficacy. Right. So we're not anticipating significant adverse events and transfusion-like responses out of this product.
Thank you for that. This is a really important question, obviously, because I want to talk about, the actual offering. What's the company's valuation at this time? And how do you see it growing once you get to your IND? How big is this offering? Can you give us some specifics?
Sure. The company valuation is at $7.5 million, which I think is a fair valuation to get us going and get us to that IND, which I think is a critical point. If you just did risk-adjusted net present value, et cetera, it would be higher. But I think this is a price point that a lot of people are comfortable with and can see good upside on their investment. What I anticipate now. Let's go back to forward-looking statements, right? No promises made here. It's always conditions in the market. But if you kind of look at, there aren't true comps in this space, which makes it a little bit more difficult. But if you look at, in general, for critical products like this at IND, you could be looking at, if it was just one of the two indications versus both of the indications going forward, you could be looking at valuations anywhere between $30 and $60 million at IND. Obviously, we're looking at price points of at the end of at least one of the phase 2 studies, anywhere between $250 to $350 million deals at that point in an acquisition or a licensing event with royalty streams attached to them. So these are sort of standard numbers. If our end of phase 1A, if our end of phase 1 study looks good in terms of alloantigens, with the bump up in overall market potential, those numbers in the phase 2 area could bump up even more significantly. And I'm trying to be relatively conservative in these estimates. Obviously, the markets drive this. Obviously, the public markets drive this as well. But my goal here is to be opportunistic and build as much shareholder value as I possibly can while we actually bring what I think is a truly breakthrough product to market.
Yes, absolutely. Couple more questions. most level 3, this one from one of our doctor attendees, most level 3 lumbar spinal infusions have less than 5 units of blood loss. The surgeon does not know ahead of time to know how much blood he will lose in order to decide to use RMP 402. Cell saver blood replaces the patient's own blood to replace blood volume to support blood pressure and enable blood oxygen transport. Does infusion of RMP 402 mean you cannot use the cell saver? Does RMP 402 support blood pressure and transport oxygen? Should all fusion patients receive RMP 402?
Good questions. RMP 402 should not affect using the cell saver. It in and of itself has no oxygen carrying capacity. It would be a very small volume. It's only about 50 milliliters administered at that point. So it would not lead to blood pressure control through most mechanisms that I can think of. Mark might have other thoughts on it. So this becomes kind of revenue neutral at 3 units. And yes, the dilemma is you don't know who is going to have that significant amount of blood loss. With the three units and above, they tend to go higher. That average unit also includes a lot of the single level or double level fusions. And, you know, at some time in the future, as I alluded to earlier, a laboratory assay could be done in looking at RMP levels of individuals. And if they're depressed, then those might be the individuals you want to give this to in advance of surgery, even though they may not have substantial risk factors otherwise for large amounts of blood loss. So I think in the case of the spinal fusion surgery, you know, one of the other advantages that we think is going to bear out is when are they losing blood? They're losing blood when they're cutting through bone. They're losing blood when they're cutting through muscle. You have a blood-filled field there that you're fighting against. Now, this is the gastroenterologist talking. This is not the surgeon talking, right? Mm-hmm. But we believe that if we can assist in helping get them a bloodless field or a substantially reduced amount of blood in the field, that will, you know, potentially increase the speed of surgery, make it easier on the surgeon in general, you know, potentially increase the accuracy of the surgery if they can accomplish that. Those are sort of intangibles that will be monitoring through the clinical trial process to see if we can capture some of that information.
Rif, I've heard it said that 50 to 60% of the surgeon's time is spent doing hemostasis.
So you know, if we can aid in that with the product, like I said, that's an intangible benefit that comes out of it. Did I answer all of the components of that question?
Yes, I believe you did.
Okay.
Thank you. So good technology and science, but you had a chart going that you had on the slide that said you were going after a lot of different bleeding conditions for use. Which ones do you think you'll focus on to get the science approved in the most effective manner?
The two are the 2 clinical trials, and it's a very good point in that we talk about the broader markets, but you know, there's marketing and there's the FDA. And if you think about it, as a company, our first customer isn't even the patient or the physician. Our first customer is the FDA. If we can't sell the FDA, we can't sell a product, right? So often I joke about the fact that the first thing I think about when I wake up in the morning with this company is, what can I do today to make the FDA's job a little bit easier to get this product approved? And the clinical slide, I think, gives you the understanding of where we're focused. Brain hemorrhage is a single study under the Orphan Drug Act that is very focused. has very concrete endpoints. That would be the size of the hemorrhage as well as functionality at 90 days. Those are pretty standard endpoints in the brain hemorrhage space at this point. In the excessive bleeding arena, it's using the spinal fusion patients as the model for excessive bleeding. For most of these patients, they don't have any other significant morbidity. We could do this study in a number of these other indications, GI bleeding, trauma, et cetera. All of those things are complicated by the fact that patients have other underlying clinical problems. In the case of spinal fusion, these tend to be normal volunteers with a back problem. Right. So it keeps your package label clean in terms of adverse events that are unrelated to your product, but it also gives you a chance to prove your product in as crystal clear a manner with quantifiable endpoints to the FDA. Those two programs can run simultaneously. They don't take much, they don't cannibalize resources in particular from each other. I have run clinical programs in cell pathways with multiple clinical programs running at the same time. So this one is not problematic for a small company, but it keeps our eye on the ball on what do we need to provide to the FDA to get them to recognize that this product is eventually approvable.
Thank you for that. So one last question. One of the last slides that you talked about some of the larger potential partners down the road. Which one of those have venture capital divisions, and are any of those planning to invest right now?
Bayer has a private equity arm that doesn't invest this early. We've had that discussion. CSL and Grifols and the other ones that are in the in the fractionated blood product space don't have venture arms. Baxter does, but haven't had real conversations there yet. I'm looking at, we're raising $3 million now. Though we can go to a larger number if there's anyone interested in that. We are open to those sorts of discussions. But I'm looking at those sort of venture arms. Of course, Novartis and the big players all have venture arms associated with them. But I'm looking at them as clinical stage. Right now we're well into this fundraising of the $3 million. We're just at about $2 million in capital in as well as committed, and we just we raised $500,000 just in the last week. Of that. So we're accelerating rapidly to get to that 3 million, but preserving most of those larger players for the larger amount of money we're going to need to get through the clinical program.
Right. Yeah, that makes sense. Okay, I think we're going to leave it there. I want to thank you, Rif, for coming on and giving us all this great information. Obviously a very exciting time for the company. If anyone wants additional information on the funding or on the company in general, if you have any questions that were not answered today, feel free to reach out to me. I can certainly put you in touch with Rif, and he'll answer any questions that you will have. I'm going to send out a feedback link to everyone who was on the call today. There's not going to be a keyword for that, so don't worry about it. But I would like to get everybody's feedback on today's meeting. And again, I want to thank Rif. Mark, do you have any parting words?
No, I just hope people really pay attention to the great opportunity put in front of you. I hope we hear from you. Seriously.
Yeah, absolutely. And thank you again, Rif, for your time. And I want to thank all of our attendees today for their time as well. I said this in other meetings. We very rarely get involved with private companies. Most of you know us as presenting publicly traded companies. So for us to get involved in a privately held company, we
It's a gem.
believe that
It's a
the
gem.
science is sound, absolutely, and it's been fully vetted by Dr. Swing, and we feel that Rif and RxMP definitely have a lot of great things happening. So I ask everyone to take a close look at this. And again, if you have any questions, feel free to reach out. So thanks again, Rif. Thanks, Mark. Until then, till the next time, we will talk to you soon. And everyone be well and be safe.
Thanks.
Thank you.